Namaste everyone, and thank you, the unfit candidate, to attend such a intellectual seminar on stem cells and future of genetics. I want to begin with a small experiment, if you don't mind. Gently close your eyes for just 10 seconds. Take one deep breath and exhale. Continue now with normal breathing. Open your eyes, please. What you just did, that tiny act of conscious inward tuning in, altered the biochemistry of this room. Measurably, verifiably, at a vibrational level, at a molecular level. What I'm sharing is not poetry. That is epigenetics. And what I'm going to propose to you today goes one step further into a territory I call endogenetics, the science and practice of using the inner environment, the landscape of consciousness itself, as a modulator of genetic expression and cellular regeneration. What the cell biologist studies under the microscope and what the meditator with his closed eyes encounters in the depths of stillness turn out to be speaking about the same reality from two different directions. Let us explore this together. The title of today's talk, Regenerative Potential through Spiritual Practice, was deliberately chosen to be provocative in the best scientific sense of the word. In this hall of stem cell science, regeneration is our shared language, but I am asking you to consider that regeneration has an inner architecture, one that has been overlooked in the excitement of cellular therapies, gene editing, and organoids. You will see four themes across the bottom of this slide: epigenetics, endogenetics, stem cell renewal, consciousness, and DNA. These four are not four separate topics. They are one integrated story, and it is the story that is unfolding in our own body right now as we sit together in this hall. At the heart of everything I want to say today is a single question, one that I have been sitting with for so many years, and one that I think science is now finally equipped to begin answering. Can what happens inside us, our thoughts, our emotions, our meditative states, rewrite how our genes speak? Not metaphorically, not as a wellness aspiration, but biochemically, epigenomically, as a matter of molecular fact. The right side of this slide gives you the three pillars of today's talk. Epigenetics is our starting point, the well-established science of how environment shapes gene expression without changing the underlying DNA sequence, genome. This is mainstream biology now. It has its own journals, its own Nobel conversations, its own clinical applications. But I want to extend the conversation into endogenetics, a term I am using to describe what happens when the inner environment, not the external, our inner environment, not the diet, not toxins, not temperature, but internal, the climate of the mind, the state of the heart, the quality of consciousness. How mind, heart, consciousness, they trigger genetic manipulations. And the destination of this journey is regeneration, the capacity of the body to renew itself. Specifically, as you all will appreciate more than most the capacity of stem cells to respond to signals that we have not yet fully catalogued. Signals that may include the very quality of our inner being. Let us begin with what we know, with the well-mapped terrain of epigenetics. The word itself comes from the Greek epi, you all know. Then is the science of what operates above and upon the genome. Here is the key insight. Your DNA sequence is more or less fixed. But the way your DNA is read, which genes are switched on, which are silenced, at what volume and at what time, is extraordinarily dynamic. And it is regulated by a set of molecular mechanisms that are breathtakingly responsive to experience. The first mechanism is DNA methylation. Methyl groups, small chemical tag CH3, attach to cytosine bases in the genome. And when they do, they effectively silence the gene beneath that. This is not random. It responds to diet, to stress, to exposures. It can be passed to daughter cells. In some cases, there is evidence it can be passed to the next generation. The second mechanism is histone modification. Think of DNA as a very long piece of thread. In order to fit inside the cell nucleus, that thread is wound around the protein spools called histones. It's a real estate efficiency. When chemical tags, for example, acetyl groups, methyl groups, phosphate groups, they are added to or removed from histones, the DNA either loosens from the spool, making it accessible for transcription, or tightens around it, rendering it inaccessible. Open chromatin means active genes. Closed chromatin means they are all silenced. Now, third mechanism, perhaps the most fascinating, is the world of non-coding RNA. For decades, the large portions of the genome that did not code for protein were dismissed as junk DNA. We now know they encode a sophisticated regulatory layer, microRNAs, long non-coding RNAs, and others. Molecules that fine-tune gene expression with extraordinary precision. These are not bystanders, they are conductors. So the genome is not a blueprint that prints the same building every time. It is an instrument, and life is the music played upon that. What are the signals that drive these epigenetic changes or even endogenetic changes? You will see 6 on this slide, and they are a good summary of what the field has established. Let me walk through them briefly and then draw your attention to the 6th. Diet and nutrition, you all know. This is one of the most thoroughly researched areas. Folate, polyphenols from berries and green tea, curcumin, sulforaphane from broccoli. All of these have demonstrated capacity to alter methylation patterns. The field of nutritional epigenomics has become a serious subdiscipline. Second is chronic stress. Here the data is sobering. Cortisol and glucosteroids, the hormones of sustained psychological stress. They have been shown to dysregulate thousands of genes simultaneously. Stress is not just a subjective experience. It leaves molecular fingerprints on the genome that in some cases can persist for years. Third factor, or rather the third modulator, is physical exercise. They activate AMPK signaling, remodels histones associated with muscles and neurological genes, and increase BDNF expression, a critical neurotropic factor. The epigenome of a person who exercises regularly, not once in a year, is measurably different from that of a sedentary person of identical genetic background. Fourth modulator, sleep quality. Specifically, deep sleep is when the circadian clock genes reset themselves and when much of the cellular repair machinery is activated. If you disturb this cycle and you disturb the epigenomic foundation of health. Fifth modulator, environmental toxins, pesticides, heavy metals, air pollutants. They all can leave lasting genetic marks. The field of environmental epigenomics is expanding these days very rapidly with profound implications for public health. Now the sixth modulator, which I like to go in detail with you all, is meditation and stillness. For a long time, this item would not have appeared on a list alongside diet and cortisol. It would have seemed too soft, too experiential, too unquantifiable. Then what changed? The data changed. And I'm going to share some of those data with you. So we need to now build a bridge between epigenetics and endogenetics. Classical epigenetics, what I am calling exo-environmental modulation, has focused on factors that come from outside the person. The food they eat, air they breathe, chemicals they encounter, the social conditions they inhabit. These are real, they are powerful and very important. But there is a second environment, equally real, arguably even more intimate, that has received far less systematic attention in the genomics literature. That is the environment that exists within the person, the emotional climate, the quality of your attention, the habitual patterns of thoughts and emotions, the depths of your meditative absorption, purpose with which you interact with individuals, with your spouse, within your family. I am proposing the term endogenetics from the Greek endon, meaning within, to describe the study of how this inner environment modulates gene expression, cellular behavior, and ultimately health and regeneration. You can see on this slide the contrast between the two columns. On the left, diet, toxins, physical environment, social conditions. On the right, thoughts and intentions. Emotional climate, meditative depth, states of consciousness, spiritual practices. Both are real, both are measurable, at least in their downstream effects. And the question this slide poses at the bottom is the question I want you all to carry through the rest of this talk. Here is the question. If the outer environment can reshape the genome, so can the inner environment. The landscape of consciousness itself. Can we make a difference there? With this slide, before we proceed further, let me define this term more carefully because I want to be rigorous rather than poetic. Endogenetics, as I am using it, proposes that the psycho-spiritual inner environment, the complex of consciousness, intentions, emotions, and contemplative states, they all act as a potent modulators of gene expression, cellular regeneration, and health. It operates through identifiable biological pathways, and it produces measurable genomic and cellular outcomes. Notice this 3-column framework on this slide. On the left, the inputs, the qualities of inner climate. In the center, the biological mechanisms by which these inputs travel into the cell. On the right, the genomic and cellular outcomes. Let me walk through the pathway. Meditative stillness, states of compassion, reduced mental noise, positive intentions. These are not vague. They have specific psychophysiological signatures. Brainwave patterns shift. Heart rate variability increases. Autonomic balance tilts toward parasympathetic dominance. These changes engage specific biological pathways. The HPA axis that you all know, hypothalamic pituitary adrenal axis, that governs the stress response, becomes less reactive. The vagal nerve, the great conductor of the parasympathetic system, becomes more active. Cytokine profiles shift. Neurotropic factors increase. And these pathway changes produce genomic outcomes. Anti-inflammatory genes are upregulated. Stress response genes are downregulated. Telomerase is activated. Cellular senescence is slowed. The stem cell niche, the microenvironment in which stem cells reside and receive their developmental signals, becomes more regenerative. This is not a metaphysical claim, it's a mechanistic pathway. And the evidence for each step in this pathway is now substantial enough that I believe it deserves our scientific attention. Now here we're going to explore stress and epigenome axis. I would like to spend a moment on the negative case because it is paradoxically the strongest argument for the positive. We know with considerable precision what chronic psychological stress does to the genome. It is not pretty and it is not reversible without active intervention. The cascade proceeds in 4 stages that you can see on the left side of this slide. Chronic psychological stress, rumination over it, the fear that grips you, the unresolved emotional weight that millions of people carry activates the HPA axis. Cortisol floods the system. This is adaptive in acute situations. It is catastrophic when sustained. The sustained cortisol leads to epigenetic dysregulation. Inflammatory genes, they are switched on. Repair genes, they are switched off. Anti-aging gene expression profile reverses. NF, KB, inflammatory pathways are activated persistently. The very pathways associated with cardiovascular disease, neurodegeneration, and metabolic syndromes. And at the end of this cascade, we see stem cell exhaustion. The stem cell niche, already compromised by inflammation and oxidative stress, loses its regenerative character. The body's capacity to renew itself is quietly and systematically diminished. Now look at the right column. What if we could reverse this cascade? What does the evidence actually show? Meditation reduces cortisol by 20 to 50%. This is a robust finding across multiple laboratories and multiple meditational modalities or practices. Loving kindness practice activates anti-inflammatory pathways. Mindfulness has also been shown to reverse stress-induced methylation patterns. Prayer, when you offer prayer, not as a business transaction to gods and goddesses, but sincere, authentic prayer, and the state of inner surrender, it calms the HPA axis. Heartfulness practice, the form of meditation I will discuss shortly, has been shown to induce coherent alpha brainwaves and significantly elevated heart rate variability. Both are the markers of parasympathetic dominance and reduced allostatic load. And at the cellular level, to bring this home to concerns of this conference, deep meditative states have been associated with telomerase activation. We'll come to the data of this shortly. But the headline is this: inner peace is not metaphor, it is medicine. It is epigenetic medicine. We turn towards the evidence. I want to highlight 4 landmark studies, each from a rigorous academic institution that together build a compelling scientific case for the endogenetic pathways. First is from Herbert Benson's group at Harvard, the landmark 2008 study on relaxation response. What Benson and his colleagues did was remarkable in its simplicity. They compared the genomic profiles of long-term meditators with non-meditators and found not just scattered differences but systematic upregulation of genes involved in energy metabolism, mitochondrial function, and insulin secretion. Crucially, they found a reversal of disease-related gene expression patterns. And this reversal was detectable within as little as 8 weeks of practicing, even amongst the novices. 8 weeks, this is clinically meaningful. The second study comes from Elizabeth Blackburn and Elisa Appel. Blackburn, as you know, was awarded Nobel Prize in 2009 for her work on telomeres and telomerase. But Appel brought to this collaboration was the psychological dimension. She was asking what psychological states and practices affect telomere length and telomerase activity. The answer was striking. Mindfulness meditation significantly increases telomerase activity. Long-term meditators have measurably longer telomeres than matched controls. The biological clock, it turns out, responds to the inner climate. The third study comes from Richard Davidson's laboratory at University of Wisconsin, one of the world's leading centers for contemplative neuroscience. In his landmark 2003 study, participants in an over 8-week mindfulness-based stress reduction program showed not only the well-known left prefrontal brainwave shift that is associated with positive affect, but significantly higher antibody titers following influenza vaccination. The immune system responded more robustly. Mind, brain, and immune gene expressions were moving together. Now the fourth study, again from Harvard, from Dusac group, performed whole genome microarray analysis on subjects before and after relaxation response practice and found something extraordinary. The genomic profile produced by relaxation practice was essentially the opposite of the genomic profile produced by chronic stress. Anti-inflammatory pathways were activated. The NF-kB pathway, one of the master regulators of inflammatory processes, was downregulated. Cellular repair pathways were upregulated. 4 studies, 4 institutions, one convergent message. The inner environment is a real and potent regulator of gene expression. We at Heartfulness also have conducted many studies on measuring the brainwave patterns, telomere length, heart conditions, blood pressure, oxidative stress reductions at multiple levels. During this session, if you all have time, one of our trainers will help you relax and meditate. You don't have to go through 8 weeks of studies. You'll be able to understand just within few minutes of this heartfulness way of meditation. I encourage one and all to try. Now we move to the slide number 9. It's all about telomeres, stem cells, and contemplative practices. I want to linger on the regenerative biology for a moment, because this, I think, is where the most exciting frontier lies with you all. Telomeres are, in a sense, the biological correlate of vitality. They are repetitive DNA sequences at the ends of chromosomes, functioning as the famous analogy that goes like the plastic tips on shoelaces, preventing the chromosomes from unraveling during cell division. Each time cell divides, telomeres shorten slightly. When they become critically short, cell enters senescence, a decaying process, aging process, a state of arrested function, or it undergoes apoptosis. Telomerase is the enzyme that rebuilds telomeres. It is expressed at high levels in stem cells and germ cells, in cells that need to divide repeatedly throughout life. It is what allows stem cells to maintain their regenerative character. And it is what spiritual practice in a growing body of evidence appears to upregulate. The numbers on the right side of this slide tell the story in a very concise way. A 43% increase in telomerase activity, BDNF, brain-derived neurotropic factor, gets upregulated amongst meditators. When you meditate, You may not just be calming your mind, you may be literally growing it. IL-6, a pro-inflammatory cytokine, and one of the most important mediators of the inflammatory microenvironment, is reduced in meditators. And this SIRT3, a longevity gene governing mitochondrial health and cell function, is upregulated during fasting combined with meditation. This connects the endogenetic pathways to the now substantial literature on caloric restriction, sirtuins, and healthy aging. The picture emerging is not of scattered, unrelated findings. It is of a coherent biological mechanism by which contemplative practice shifts the cellular landscape toward regeneration. Now, heartfulness that I've been practicing since 1976, and millions of people who are practicing today. And I request you all, please try it at least once in your lifetime. At least once. And see if that's a cup of tea you like to consume. I want to now speak in detail from my personal background, from the tradition of practice that I know most intimately, as an illustration, not an exclusion. Every authentic contemplative tradition, be it Zen, be it Sufism, be it Vedantic, or Carmelite Christian, or even Theravada Buddhist tradition, they all may offer its own version of this endogenetic pathways. What I am describing here applies wherever genuine depth of practice is attained. The heartfulness tradition, rooted in Sahajmarg lineage, offers what I believe is one of the most complete and practically accessible models of endogenetic practice available today. It is practiced by millions of people across 160 countries, and it is increasingly the subject of rigorous scientific studies. The practice has 4 pillars, and each has a specific biological relevance. First is relaxation. We have already seen the effect, scientific evidence of what relaxation could do at a cellular level and how it changes inner climate. A systematic settling of the body and mind before meditation. This is not generic relaxation. It's a progressive descent into stillness that activates the parasympathetic nervous system. It lowers cortisol and creates the psychophysiological conditions for the inner environment to begin shifting. Second element is meditation. Heart-centered awareness combined with unique element of pranahuti or yogic transmission. This is a topic that deserves its own scientific investigation actually. The idea that a trained practitioner can transmit a refined state of consciousness to another person. There is preliminary evidence from EEG studies showing coherent brainwave synchrony between a trainer and student during transmission. The third is cleaning. An evening practice in which the practitioner consciously releases the accumulated impressions, the baggage. The baggage, I would call it as emotional baggage as a result of interacting with the world. Knowingly or unknowingly, it builds up something within us. From an endogenetic perspective, this is particularly interesting. If chronic emotional residue is one of the primary sources of sustained stress that drives epigenetic dysregulation, then a daily practice designed specifically to dissolve the residue is epigenetic dose of medicine in the most literal sense. The fourth is prayer, or what in this tradition we call it as inner communion, a state of deep surrender and connection. This activates what neuroscientists call default mode network, DMN, in a regulated restorative modality, quite different from ruminative activation of the default mode network, that is associated with anxiety and depression. Now, I want to bring this to the practical level, because this is where science and daily life must eventually meet. This particular slide shows a timeline from early morning, dawn to night of what an endogenetically informed individual whose practices will look like. I am not presenting this as the only way, though I would like that you all embrace this. I am presenting it as a structured example of how the findings we have discussed can be embodied in our daily life and make it our daily experience. At dawn, the morning meditation. If you can't, any other time is good enough. Research on cortisol awakening response, the spike in cortisol that occurs naturally in the first 30 minutes after waking, shows that this is one of the most epigenetically sensitive moments of the day. Beginning the day with meditation rather than with anxious planning or screen-based stimulation sets the HPA axis, its baseline for the entire day. Through the morning intentional awareness, the practice of remaining inwardly attentive, even as one engages with work and relationships. This sustained alpha theta brainwave state is associated with BDNF release and neural stem cell nourishment. At midday, a brief centering pause, a few seconds, few moments, 5 minutes maybe, a conscious stillness triggers vagal activation and reduces the accumulation of inflammatory cytokine load. Take a pause every now and then. Go inward, become still, center yourself. In the evening, the cleaning practice. During this evening cleaning, the deliberate release of the day's psychological residue. This is the endogenetic equivalent of removing the day's toxic exposures before they become embedded in the epigenome. And at night, the bedtime prayer or moment of surrender, which promotes restorative sleep by activating the growth hormone secretion and cellular repair cycles that are bound to occur during deep sleep. Taken all this together, this is a comprehensive endogenetic protocol. It is free. It has no side effects. It is available to every human being on earth, and the biology behind it is too sound to believe. Let me close with 3 takeaways from this final slide and then with a personal reflection. First and foremost, science validates wisdom. Epigenetics is, in a profound sense, molecular confirmation of what contemplative traditions have always insisted, that how we live inside matters. What the quality of our consciousness is not irrelevant to the health of our cells. This is not a metaphor. This is molecular biology. Second, endogenetics is a clinical frontier. The next phase of regenerative medicine, I believe, will be integrative in a deeper sense than we have yet imagined. It will not merely combine conventional treatment with lifestyle medicine. It will incorporate consciousness-based interventions alongside stem cell therapy, gene therapy, and pharmacology. The inner environment will be considered as a legitimate parameter of clinical care. Thirdly, every person is a laboratory. This may be the most democratizing finding in all of medicine. You do not need access to an expensive treatment center. You do not need a personalized genomics report. You need silence. You need stillness. You need the simple, radical act of turning the attention inward and holding it long enough for the biology to respond. I'll close with the line at the bottom of this slide, which comes not from genomics paper, but from the experiential core of the tradition I follow. I have described it today. The body is the outer expression of the soul. Heal the soul, the body follows. Today, I think we are beginning to understand at the molecular level exactly what that means. Thank you. Namaste to all.